Cell biology is a fascinating discipline that unravels the complex machinery and structures within cells. Understanding how cellular components function and interact is crucial for comprehending human health and disease. In this script, we will delve into various techniques used in cell biology to isolate and study specific cellular components. Through techniques like differential centrifugation, density gradient centrifugation, and fractionation methods, researchers can separate and analyze different organelles and macromolecules. This script aims to provide a detailed exploration of 12 different techniques commonly used in cell biology research. So let's begin our journey into the fascinating world of cellular fractionation and isolation. Differential centrifugation is a method that allows researchers to separate cellular components based on their size and density. The process involves a series of consecutive centrifugation steps at increasing speeds. During each centrifugation step, different cellular components pellet out at different speeds, thus allowing their isolation. By carefully controlling the speed and duration of each centrifugation step, scientists can obtain enriched fractions of organelles such as the nucleus, mitochondria, and endoplasmic reticulum. Density gradient centrifugation is another technique used to separate cellular components based on their density. This method involves layering a sample onto a density gradient, typically a sucrose or cesium chloride gradient, and subjecting it to ultra-high centrifugal forces. As the gradient spins, particles of different densities move through the gradient until they reach a region where their density matches. This results in the formation of distinct bands containing different cellular components. Density gradient centrifugation is particularly useful for isolating membrane fractions and studying their composition. Isolating the nucleus is a critical step in studying nuclear processes and activities. The process involves disrupting the cell membrane and subsequent differential centrifugation steps to separate the nucleus from other cellular components. Techniques like homogenization, density gradient centrifugation, and filtration are employed to obtain highly pure and intact nuclei. Isolated nuclei can then be used to investigate transcriptional regulation, DNA replication, and other nuclear functions. Mitochondria are vital organelles involved in energy production and numerous cellular processes. Isolating and studying mitochondria can provide valuable insights into their functions and dysfunctions. Techniques like differential centrifugation, density gradient centrifugation, and selective disruption of other organelles are used to obtain pure mitochondrial fractions. These isolated mitochondria can be further characterized by assessing their enzymatic activities, membrane potential, and morphology. The endoplasmic reticulum, ER, is an intricate network of membranes involved in protein synthesis, lipid metabolism, and calcium homeostasis. Isolating the ER allows researchers to study its structural and functional properties. Techniques like differential centrifugation, density gradient centrifugation, and selective disruption of other organelles are employed to obtain enriched fractions of ER membranes. These isolated ER fractions can be analyzed for enzyme activities, protein composition, and membrane structure. The Golgi apparatus is a crucial organelle involved in protein sorting, modification, and transport. Isolating and studying the Golgi apparatus can shed light on its role in cellular processes and disease mechanisms. Techniques such as density gradient centrifugation, immunoprecipitation, and immunoelectron microscopy are employed to fractionate and identify Golgi membranes. These purified Golgi fractions can then be used to study vesicle trafficking, glycosylation, and Golgi-related diseases. Lysosomes are essential organelles involved in the degradation of various cellular components. Isolating and studying lysosomes can help unravel their role in autophagy, phagocytosis, and lysosomal storage disorders. Techniques like differential centrifugation, ultrafiltration, and density gradient centrifugation are used to enrich lysosomal fractions. These isolated lysosomes can be characterized by analyzing their enzymatic activities, membrane composition, and protein trafficking pathways. Chloroplasts are unique organelles present in plant cells, responsible for photosynthesis and the production of carbohydrates. Isolating and studying chloroplasts is crucial for understanding plant metabolism and responses to environmental stimuli. Techniques such as differential centrifugation, using osmotic shock to rupture other organelles, and plastid-specific markers are employed to obtain purified chloroplast fractions. These isolated chloroplasts can be further analyzed for pigment content, photosynthetic activities, and protein localization. The cytoskeleton is a dynamic network of proteins that provide structural support and regulates cellular movement. Isolating specific cytoskeletal components allows researchers to study their functions in cell motility, division, and signaling. Techniques like differential centrifugation, selective depolymerization, and immunoprecipitation are used to isolate distinct cytoskeletal elements such as microtubules, actin filaments, and intermediate filaments. These isolated cytoskeletal fractions can then be analyzed for protein composition, post-translational modifications, and interaction partners. Cellular membranes play critical roles in compartmentalization, signaling, and transport processes. Isolating and analyzing specific membrane fractions allows researchers to investigate their lipid composition, protein composition, and functional properties. Techniques like differential centrifugation, membrane flotation, and proteomic analyses are employed to obtain enriched fractions of plasma membrane, endosomal membranes, and other specialized membrane compartments. Studying the protein synthesis machinery is essential for understanding the regulation of gene expression and protein synthesis. Isolation of ribosomes, polysomes, and translation factors allows researchers to investigate translation mechanisms and their dysregulation in disease. Techniques like differential centrifugation, sucrose gradient centrifugation, and immunoprecipitation are used to fractionate and analyze the different components of the protein synthesis machinery. Nuclear pore complexes, NPCs, are complex protein assemblies that control the molecular exchange between the nucleus and cytoplasm. Isolating and studying NPCs provides insights into the mechanisms of nucleocytoplasmic transport and their role in cellular processes. Techniques such as differential centrifugation, digitonin permeabilization, and immunoprecipitation are employed to isolate purified nuclear pore complexes. These isolated NPCs can be characterized by electron microscopy, mass spectrometry, and functional assays. Cell fractionation and isolation methods play a crucial role in cell biology research. These techniques allow scientists to separate and study different cellular components in detail. By investigating organelles, membranes, cytoskeletal elements, and protein complexes, researchers gain insights into their structure, function, and involvement in diseases. These methods bring us closer to understanding the complexity of life.